Good afternoon, everyone. It's my pleasure to welcome you to the WIGGIS Workshop. As you know, the topic of the event is on the integration of genomic and geographic information system data for the conservation of wildlife. But we will tackle this topic also through a number of case studies dealing with different types of species so that we can fully exploit all the types of application that these methods have experienced so far. As you probably know, this event has been made possible thanks to the funding we received from the EU in the form of the LIFE GLOW project, which is within the LIFE project framework. And so this is one of the dissemination events that are part of the project plan. I leave the floor to the first speaker, Professor Stéphane Just from the École Polytechnique Fédérale of Lausanne for one of the main lectures of today.
Thank you very much, Licia, for the invitation, first of course, for the opportunity to also talk about geographic information systems that are rarely put just at the center of the attention. And okay, it's an opportunity to describe exactly into more detail the role of these tools. So I will start this talk just with a small remark as regard the title, the wording of the title. So there is a story about the geographic information systems that I mentioned just a few seconds ago. But when I read this, I wondered if it was in fact the first workshop on landscape genomics, but without mentioning this term, in fact, because it's not mentioned in the title. It's not mentioned in the description of the workshop, in fact, that we see here. So I provided the organizers with a title that fitted this wording. So it's not present in my title neither, but finally, a few days ago, I just added a subtitle. This is some landmarks and historical anecdotes on the way to landscape genomics. In fact, this is just to make more explicit the thread of my talk. And during this talk, what I will try to do is to try to show, in fact, the importance of geographic information, the geographic approach, to analyze genomic data. And not only, because this is often the case, not only focusing on this role of integrating data or of managing the data and visualizing the data. So what we are interested in fact, this is the fact to compare information on the living organism with information describing landscapes. So the interest here, which is translated in this present in the current workshop with the word integration. This is how can we compare these two dimensions, and thus I just initially focused on early works that did the same. And among these early works, one can mention what de Candolle and Wallace, which are well-known naturalists, and they worked just to try to identify differences in community composition, patterns in the spatial distribution of species across the landscape. And they did observation to start explaining what are the physical causes that may be at the origin of these differences. And in particular, you may know this famous observation of Wallace in the Malay archipelago. This is where he observed or noted this famous biogeographic boundary in the fauna between the Australian region and the Oriental region in this area. And I make the story very short here. This is just a boundary between certain species that exist in Asia only, like the tiger, for instance, and certain species that exist in Australia only, like marsupials, for instance. And then he observed that this boundary was surrounded by a kind of transitional zone around the boundary, where you had both terrestrial and marine species also that exist on both sides. And Wallace named this type of investigation. He named it zoological geography. And what's important to us, to our point of interest today, in fact, this is that these are really early research that investigate these relationships between geo-ecological conditions and the presence or the absence of species. So what was missing here with these works, of course, this is genetic information. Genetic information, we tackle this issue. We have to consider this information, of course, today. And if we consider genetic information, there is an important step in the history of what we study today, which is the role of Wright's isolation by distance concept. And this appeared in 1943. So in this case, gene frequency is considered. And also of importance is what Wright introduces, which is the theoretical concept of the spatial variation in of neutral trace. This is really important. Then, after these milestones, we had important works conducted during the 60s. And during the 60s, clients started to be used as experimental designs, in fact. Scientists established setups to study local adaptation and simultaneously gene flows between populations. And there are also interesting works by Kostas Krimbas in Greece that designed a study around this Mount Parnes that we see here on the slide, on a natural population of flies of Drosophila subobscura. So he did previous studies where he highlighted decline in the spatial distribution of chromosomal polymorphism, and he could relate these differences, these declines, with temperature and humidity. So he was looking for this client to replicate this discovery here in Parnas. And he did several analyses during 2 years just to follow a population in this area, focusing on temperature and humidity variation. But finally, he had to conclude that, okay, temperature or dryness finally did not affect very much the polymorphism of interest. So he had to finally reject his hypothesis. But what was interesting, or what is interesting for us in this study, is that we have all the ingredients of the analysis of the relationship between frequency of genetic features. So here, this is frequency of the support of the genetic information and the variation of environmental characteristics. But these elements are here present. Then shortly after Krimbas' works, there is a team. Pronto? Is it okay? Okay. So just after these works, there is a team in North Carolina State University that start doing very interesting analysis for us. In particular, there is a scientist named Henry Schaffer, and he analyzed with his colleagues allelic isozyme systems in a population of ants in Texas. And the approach they use is also very modern. So this is something we know and we use a lot nowadays. This is principal component analysis. And you use this type of analysis to compare the variability of allele frequencies with environmental factors. And they obtain significant correlations. And they discovered that some climatic or weather variables, like they named them, were correlated with these allelic frequencies. So they thus suspected that natural selection was really important in determining the pattern of these allele frequencies. And like Grimbus in Greece, again, with this type of analysis, we have all the ingredients of what we are doing now and what we are studying now. So this is relationship between frequency of genetic information and a measured variation in environmental parameters. And the same team conducted a bit later similar studies, but on flies, on Drosophila melanogaster this time, to investigate the relationship between gene frequencies and geographic locations. And this time, what is really interesting, and one may say anticipated, is the fact that they took into account the issue of spatial autocorrelation. So it means that globally, I will come on this concept later, but with more details. But here, this is in fact, you have the replication of the same behavior locally. So it may bias the results of the association analysis, but in fact, they correct for this bias. And despite they have to implement this, in fact, they still find significant correlations between genetic and environmental variables. And also an important element with this study is that they deepen the analysis and the use of a variety of environmental predictors. They used wind speed, for instance, or relative humidity at different times of the day. So the temporal scale was also investigated, what makes this study also very interesting. And this interest for the variation in environmental information and also this interest for the role that this environmental information is playing in shaping or in driving genetic variation in the organism. This is an element which is really fundamental to molecular ecology. And molecular ecology, this is a field pioneered by Godfrey Hewitt. And what Godfrey Hewitt did was really to place this genetic diversity in a spatial temporal context. And he also showed how to integrate landscape ecology and the history of the studied species through phylogeography. So this is important because this field of molecular ecology, this is the very discipline in which landscape genetics and landscape genomics, we will discuss a bit later, have their roots, in fact. So what I show here, this is a case study, a paper written by Godfrey Hewitt. This is an example dedicated to grasshoppers, a grasshopper species in the south of France. And in his work, analyzing the genetic data and the spatial distribution of the genetic data, in fact, Godfrey was able to identify two different populations with different chromosome types separated by geographic barriers. So he highlighted these geographic barriers. And he highlighted also hybrid zones with hybrid populations, and had a theoretical hypothesis or postulations to present how this kind of process may occur in immobile or almost immobile species that are, in fact, due to this immobility, subjected to isolation. And then they are subjected also possibly to expansion, depending on the climatic and ecological changes that may happen, like in particular, and this is also very central to the work of Godfrey Hewitt, the consequences of the Europe's glacial history. And another element really important and really interesting in the work of Godfrey Hewitt, this is the way he designed the first map with a geographic and environmental context, like you can see here. These maps start giving an idea that can be visualized about the landscape, about elevation, and this is something with the context, with the location of the rivers, etc. And this is an important contribution also. And just after this interesting and important works of Godfrey Hewitt, there was a very rich review paper written by Hedrick and several colleagues, dedicated to genetic environment association studies. And this paper gives an idea of the variety of the many studies that investigated these relationships between genetic polymorphism and environmental variables. But at this moment, it's 1976. And this review in particular, it contains a reference, a well-known reference. This is a famous paper of Bishop, where he investigates, in fact, a decline in the melanic frequency in most species between Liverpool and the rural part of Wales. Investigates this decline. In fact, you have this species of moth, which is initially white and which is living on the birch trees, but then gradually, because of air pollution and due to the exploitation of coal in particular, the tree became black, and in fact, gradually also, gradually the moth also became black, just to remain invisible to predators. So this is a perfect example of natural selection. So this is 1976. And just after, interesting works by Jeffrey Meaton and different colleagues on pines. And they did many different studies in the context of which they correlated allelic frequencies with a few environmental parameters. This is mainly related to elevation, and different aspects. And they are able to detect a few associations between allelic frequencies and slopes or different aspects. They discover also that an excess of heterozygosity is associated with xeric habitats, and they also work on the angel spruce a bit later, and very clearly highlight the importance of soil moisture to be involved in selection processes. And we reach the late 1980s, and during this period, there are two important articles that are really at the center of what we are discussing today. And this is this process of integrating geo-environmental and genetic information. So the first paper contains the recognition of the importance of the interaction between the genome and the environment. This in order to better understand evolution. And this paper, it was written by Robert Berry, and at this time he was president of the British Ecological Society. And this paper influenced the title of my contribution today. This is where genes and geography meet. And a bit later, there was a second paper by Jeliński, in which he also highlights the necessity to connect genetics and ecology, with a title also referring to genes and geography. So this interaction and the connection which is requested by these two papers here could be performed at this moment by a new type of computer tools that appeared during the 1980s. And these tools, this is the first personal computer. So we had the evolution of computer science, And what we see here, these personal computers appear, and then simultaneously you have the gradual development of geographic information systems, so at the level of the software. And the combination of the two created this place where the genes and landscape meet, but in modeling terms, and these are in fact geographic information systems. And these tools, GIS, they are really key to landscape genomics, because one of the main functions, so again, beyond only the management and the display of geographic information, this is really this capacity to overlay different layers of georeference information, like genetic data, environmental data. But this overlay function, it materializes, in fact, the connection which is necessary to establish this link between genetic and environmental information. And this overlay function, and there is in geographic information science a concept associated which is named spatial coincidence. This is what we see here on the slide. Constitute this the articulation that make it possible then to elaborate association models in landscape. Now, I remain on this thread and temporal thread. So we move to 2003, and 2003, this is the year when this paper by Stéphanie Manel, Mike Schwartz, Gordon Leuckart, and Pierre Tabelin was published. And this is the first paper referring and defining the term landscape genetics. And in this paper, in fact, there is a clear reference to geographic information systems. This is the section I highlighted here on the slide. The paper, confirms the important role of these tools to visualize spatial patterns and overlay different types of useful reference information. And this is what is written, and this is also the reason why then this talk will move also and stress also other functionalities, functions of geographic information systems. This paper also mentions interesting functions like the correlation between different environmental layers, and it has mainly an important quality. This is that it increases the awareness for of these important effects that spatial patterns of landscape elements of environmental factors have on neutral and adaptive genetic variation. And the paper has another merit. This is to federate, in fact, a research community, because just before this paper, all the different works that I have presented, described, they are dispersed among different fields related to evolutionary biology. And then finally, this paper is important because combined with other papers that I will present now, it constitutes, in fact, a part of a puzzle that gradually led to the emergence of landscape genomics. And the second piece of the puzzle is a paper that was published the same year, and which is about population genomics. It was published in Nature Reviews Genetics, and this paper describes a protocol in several steps on how to implement population genomics. It explains the potential of this discipline, and it explains also how it would become possible to take advantage of high throughput sequencing technologies. And in this paper, the first author is Gordon Leuckart, so he was also author of the paper I presented before. And the senior author of this paper is Pierre Tabelin, also the senior author of the paper mentioned before. And if you go to the references of this paper, this is what you see. In fact, what Pierre Tabelin did is to comment the references, and he comments the paper by Stéphanie Manel on landscape genetics. And he says here that he explained, in fact, the potential of this new discipline relying on the advent of high-throughput sequencing technologies. Explain that if you combine the two, the present paper and the paper on landscape genetics, in fact, we may reach an approach one could call landscape genomics, using association studies between the genome and the environment. It's a kind of anticipation of what could take place afterwards. And, during this time, in fact, there is a European project, sorry, we are here, named Econogen, whose research goal is, in fact, to study the conservation of how to conserve small ruminant genetic resources in Europe. This is an interdisciplinary project. This project is chaired and coordinated by Paolo Raimone. And in the team, we also have Godfrey Hewitt, Pierre Taballet is present also, and Mike Burford also. And during this project, this is a reflection phase, in fact, related to this remark of Pierre Taballet on how can we combine both approaches, how can we do it, and this was one of the goals of this project. And finally, after the end of the project, we were able, because I had the chance, in fact, I did my PhD in the context of this research project, and one of the goals was to find a way to combine these two different types of information in order to be able to provide an approach that could be regularly implemented with software tools, etc. So this is what we did here with a paper applied to sheep breeds, but also pine weevils, and in fact, we were able to produce a software able to process these association models. So you can see here on the slide that we used a very few numbers of molecular markers and environmental parameters compared to what we are doing today, of course. But okay, the framework was here and ready to evolve because, in fact, very soon, because of these large data sets and molecular data sets in particular that were soon arriving, it would be necessary to compute much more processing load, in fact. So the software, it was necessary that such a software would be able to calculate billions of models, hundreds of environmental variables, and okay, the ingredients were here. And then I had a PhD student, Sylvie Stucki, and she developed a C++ version of the software. And a bit later, we produced also a version that made it possible to drive or conduct the analysis from R. This is R Sambada. This is the current name of the application. So this is the application out of this pipeline here with the Econogen project. But in fact, there are many other solutions that exist today that do the same type of analysis. You have BayEnv, for instance. This is the work of Graham Coop in the United States. LFMM and Léa, developed by Olivier François, Eric Frichot. In Grenoble, you have GLMMM, BayPass, Basecamp, and this is the work of De Villemeret and Gadgety. And also now what we observe, this is the emergence of landscape genomics carried out by means of redundancy analysis, RDA, and this is the work of Brenna Forrester and Thibaut Kaplan. So there are many different solutions, and Elia, in his talk in a moment, will come with more details on some of this approach. My work here, or my task here, is to focus rather on our solution, because the our Sambada solution focuses or gives more opportunities to analyze the geographic dimension of the data that we analyze. And these, in particular, an important function, this is the analysis of spatial autocorrelation of spatial dependence. Once we have a variable of interest, like the frequency of a given a marker, it's interesting to see how the frequency is distributed on a territory. And this is what we say. So this is to assess the spatial dependence of allele frequency. So spatial dependence, this is illustrated by the two artificial territories we see here. So on the left, there is a situation with a random distribution of this allelic frequency. And on the right, you see that there is a spatial dependence. It means that at a specific location, the frequency is higher. So it means that there is potentially an explanation at this place for explaining this higher allelic frequency. So here is an example in a plant species, Campanula barbata. And what we did is to, in this case, it was to analyze AFLP data. And we use here a spatial statistics. This is a local Moran's I. In fact, it computes the resemblance between what we observe, a frequency we observe locally, and an allelic frequency we observed in the individual populations situated around in a given distance, in a given neighborhood, that we say. So, and when the individuals look like each other, they are classified in the red class. When they don't look like each other, it's in the blue class. And you have different types of behavior, the other classes that I won't describe here. Another class which is important, this is a white one that you see here. And this white category or class, this is where there is no spatial dependence. It means that, in fact, the distribution is random and there is no resemblance between individuals. And in fact, these white areas that we can see here on the maps, they highlight hybrid zones, contact zones between lineages that diverge. So this is confirmed by other studies, but it makes it possible to visualize this type of hybrid zones using spatial statistics, in fact. Here is another example to show how we can use this concept of or these tools, these specific statistics specific to the analysis of geographic information. In this case, we also worked, these are data produced by the Econogene project, and we also worked on the frequency of AFLP bands. And in this case, you see on the left, this is a relationship of specific markers with precipitation. And on the right, you see the corresponding map. So we mapped this environmental variable. So you see that when we move to the blue or purple color, we have more precipitation. And simultaneously, we observe a cluster, so a group of individuals, in this case, that look like the neighborhood, and they are all grouped in these zones where the temperature, the precipitation are higher. So it means that there is probably a relationship between this marker and a function in the same genomic regions, which is related to precipitation or humidity. So what is important here is just to document the fact that with this kind of association studies, it's possible to document a possible function related to the environmental variable which is associated, and that may support further investigation related to functional genomics, for instance. Here is another example in Uganda. Here we worked with whole genome data. It was in the context of another European project, the NextGen project. And in this case, we observe on the right a map also showing a local indicator of spatial association or spatial autocorrelation. And we see that in the north west of Uganda, we have a cluster with high frequencies of a given marker, of a given SNP, which is associated with an environmental variable, which is the stability of temperature, in fact. And these signatures, which is clearly, really clear on the territory, on the map here, it corresponds to a zone, a specific zone where it is known that we observe two different species of tsetse fly, of course, associated with trypanosomiasis. So in this case, we can also document the fact that a specific region of the genome is associated with a particular environmental variable. And with the comparison of the information of other studies, it may also lead to interesting information when we are studying the different pathway leading to resistance against a specific disease. And now I will also mention a more concrete role of this type of approach, a way towards real application in conservation. And the idea, I will very roughly present it here because Elia Vaiana will also give more detail about this type of approach. But this is just to explain the idea that when we compute these association models between the presence or absence of SNP or a given molecular marker and the variation of an environmental variable, like here on the plot, you have the probability of presence of a given marker varying according to the number of wet days. And we highlight a point, this is the red point here, where we see that we have a probability of 60% of having 12 days, 12 rainy days per year. And if this relationship is of interest, we can map it. And this is what we see on the two maps that we see at the bottom of the slide. We can map, in fact, the probability of presence of this marker according to the value of the environmental parameter we are using. And in this case, we use environmental information produced by different producers or different offices, environmental offices, freely available data. And using this computation, it's possible to map these area. This is what we name these geographic areas of allele frequencies or spatial area of genotype probability. And this is areas that are drawn on a map and inside which we know that we have this probability defined by the threshold. So it may be another one. This is an example with 60%, and in which we know that a given genetic variant will be useful or will miss, it's the contrary, a given specific environmental conditions. And using this type of information, using also this information given the evolution and environmental parameters, because thanks to the IPCC panel, they produce scenarios of global warming with the evolution of the different variables in time. So we can use scenarios on future conditions, redo the same calculations, and this is the difference between the two maps here, 2015 and 2017. We can observe a difference given the evolution of the environmental parameters and forecast where a given variant will be necessary to cope with specific environmental conditions. So it gives the opportunity to really draw on a map, for instance, conservation areas, given the characteristics, genetic characteristics of species that we need to conserve. But you will have more information soon. And also tomorrow, Oliver, Moni will explain how this concept, how this approach can be used also to conserve coral reefs, with the same idea of also of designing marine protection areas, for instance. But this will be for tomorrow. And finally, just before concluding, there is also another area of interest in the way we investigate the data, in the way we design these relationships between genetic and environmental information. This is, of course, we hear a lot about artificial intelligence and techniques like machine learning. And machine learning for landscape genomics, this is something which is not very often applied, but which is really feasible. The example that I show here, this is not about genetic information. This is an example on the, in fact, a combination of present absence of ticks, a tick species on the territory of Switzerland, in which we found chlamydia bacteria with specific public health problems related to what chlamydia can transmit as a disease. And in fact, what we do is that these algorithms, like maximum entropy algorithm in this case, used with a software named Maxent, it's possible to learn, for the algorithm to learn what is the environmental composition, in fact, where we observe the presence of a given genetic variant, for instance. And this can be used, of course, also to make these investigations to detect, identify genomic regions under natural selection. And in fact, this approach is very much used in with GWAS to detect and to study gene-gene interactions and not gene environment association, as it could be the case with landscape genomics. So I reach my final, my conclusive slide. The different elements I want to stress and that are interesting to keep in mind after this presentation, that okay, it's a long time that okay, we are interested in the relationships between the environment and the presence or absence of species, the presence or absence of particular genetic features also. And also compared to this story with this paper of Stephanie Manel and colleagues in 2003, this is important to keep in mind that this is well before 2003 that scientists started to do this kind of study. So the difference is that the scale of the studies was really reduced, but the ideas were already implemented, and very interesting discoveries took place at this moment. Then, and this is directly related, of course, all the progress that we did afterwards in this domain, they are narrowly related with computer science, with the progresses of computer science. There is a hardware dimension, because suddenly we had these personal computers available, and on these personal computers, more people were working, and more people were using this emerging software named Geographic Information Systems. And these GIS, then they favored the collection, they favored the availability of environmental data. And it was then possible to start correlating a few molecular markers against one or two environmental variables. And then in turn, we had the progresses in molecular biology toward the advent of high throughput sequencing technologies. And of course, and then the availability of millions of SNPs that made it possible to really implement what we name landscape genomics today. So it's the fact to run billions of association models between genetic features and environmental variation. But, and this is my final remark, the interest in GIS goes well beyond this integrative function, well beyond this visualization function. And I think that landscape genomics takes another dimension. Once the results that we produce are analyzed first by means of spatial statistics, and it's really not often carried out. There is a lot of room there for additional analysis of genetic information distributed on a territory. And then there is also an important and another dimension of landscape genomics as regards its utility when we use them on data with a spatial temporal perspective or spatial temporal dimension, and it gives this perspective to reveal the evolutionary dynamics of adaptation. And this is really important. This is a key information to consider when we start deploying and developing conservation programs. So that's it. Thank you very much for your attention.
Okay, thank you, Stefan. We have time for a few questions. Maybe if the audience is too shy, maybe I have a small question. Let's see if I can formulate it correctly. So I'm very interested in that approach of prediction of allele frequency location. Let's say it was called geographic area of allele frequency. And so I was wondering, of course, if you want to predict in the future the presence or absence of an allele, the individuals must be there. And so would it be possible to test or to study this type of constraint? Because it's like the difference between the potential niche, realized niche when we do species distribution model. I don't know if, so.
Well, this is exactly a type of study design that we try to implement. So it's planned. So we have a project now that's started on crawl species, and in the context of which, so we will do this type of investigations, and afterwards, it's planned to do experiments in aquariums to go in the areas where we did not sample, and we will sample afterwards, and then take these individuals, do the experiments in aquarium, and then check if this adaptive function that we predicted to exist is real. But until now, I don't know if you know Elia or Oliver, I don't think that this kind of validation was carried out. Mm-hmm. No, but it's on the way.
Let's see if there are, yes.
Thank you. Probably there is place for merging these two approaches. I mean, landscape genomics, spatial area of genotype probabilities, and species distribution models together. I don't think this is done, but probably it would be a good new avenue for research to really combine these approaches.
Okay.
I have another question, Stefan. So we saw during the last years that the genomic information is becoming more and more accurate and more precise, and we can have information on the genomic resolution is getting smaller and smaller. Can you see an equivalent on the environmental side? For example, what's the role of the spatial scale when it comes to adaptation? Is this something we should take into account when we study local adaptation, and how?
Mm-hmm. Yes, in fact, in this domain, we were also in the course of conducting research that would probably provide answers. There are two answers. First, there is a general increased availability of high resolution data set with the remote sensing technologies. Of course, the resolution of the sensors is improved and is more and more available and publicly available. And even if you want really precise images, in fact, you can get them. It's necessary to pay a certain amount of money to get this data, but they are available with images with incredible spatial resolution to 10 centimeters, more or less, from a satellite. So it's really increasing with a lot of different wavelengths available to do your measurements. Then there are also other sensors that make it possible to investigate adaptation process, for instance, at the local scale. And for instance, we used drones to produce digital elevation models. So very high resolution digital elevation models. It means that we reach a spatial resolution of 5 centimeters. And then what we did, so it's a study on a plant species, Arabis alpina, in the Alps, and we produced different environmental data sets with a different spatial resolution. So using digital elevation models, you can produce synthetic data sets. In fact, you can derive the information of this digital elevation model. There are several variables that you very well know. I mentioned just before, so it's aspect, it's slope, but you can also produce measure of terrain ruggedness. You can also produce proxies to soil humidity, etc., soil radiation. You can produce a lot of environmental information compared to genetic information that you will have collected. So and now the calculations that we are doing to analyze this relationship between the presence of certain SNPs of interest and environmental information, what they show is that in fact the different environmental predictors show different selective processes, but at different scales. It means that when you use a vector ruggedness measure, for instance, an indication of rugosity, in your association models, you see that you reach the highest significance for a spatial resolution of 2 meters, for instance. When you use a total solar radiation, you know that this is a model with a spatial resolution of 16 meters, etc., etc. So what the research is on the way to or able to produce as information now, and very precious information, I think so, that is to say, okay, if you want to detect something related to this or this environmental variables or this type of environmental pressure, use this variable, but at this spatial resolution. We are able now to globally, this is to say to determine at which scale a natural selection operates for a specific environmental pressure. But it means that this is not the best spatial resolution, the finest spatial resolution that will provide the best results. And this is important to the field. Mm-hmm.
Thank you. That's fantastic. I think we have to move on to the next speaker. Thank you very much. And so next speaker of this first session is Elia Vaiana from the Institute of Bioscience and Bioresources of the National Research Council of Italy. And the title of his presentation is Harnessing Landscape Genomics to Detect Signatures of Spatially Divergent Selection in Terrestrial Species.
So thank you, Marco, for your introduction, and Lisa for your invitation. I'm very honored to be here. Thank you very much. So in this presentation, what I'm going to do is just to deepen a bit this approach that Stefan introduced during his presentation, which is landscape genomics. I will go through a brief introduction where I will focus the attention on one important concept, which is related to landscape genomics, which is the one of local adaptation, in fact. So we'll describe a bit of detail what we actually mean when we talk about local adaptation and why it is related to landscape genomics. Then we will go through some applications, recent application of landscape genomics in the field of conservation genomics, and then finally do some final remarks. So local adaptation. Local adaptation, in fact, is a very important topic in evolutionary biology. Is of great actuality, in fact, because of all the problems that we are seeing with this climate change or this environmental change. In fact, local adaptation is a very widespread evolutionary response across let's say, the tree of life. We have example from starting really from viruses. We have evidence that some viruses show local adaptation until to major organisms like marine organisms, corals, or plants or animals. So why it is so important, in fact, local adaptation? Because in fact, it is that response, the specific response that allows to survive variability in natural conditions. So if you look at this map on the right of the slide, we see the spatial distribution of a species. This is, by the way, a conifer species, and we see that if we are able to characterize the environmental characteristics along this across this distribution, we have variability. Okay, so we have environmental variability if you look at this at the ecological niche of a species, right? We can find some areas that are, for instance, cold and dry, some others that are hot and dry, and so on. But the species can survive all these characteristics. So local adaptation is the evolutionary response, in fact, that allows species to respond, to tackle this variability. It's something that, of course, involves two populations or more of the same species. Two population that evolves, of course, in contrasting habitats. So if you look at this picture, for instance, we can see that we have, for instance, two populations, one, let's say, mainland and one on top of the mountain along an environmental gradient. So this population, in fact, evolves in contrasting habitats. They can be connected, they are connected by gene flow. So the exchange, in fact, the exchange genes. The fact that they evolve in these contrasting habitats is the nature of this contrasting habitat is due to, in fact, to the action of what we call spatially divergent natural selection. So natural selection, divergent natural selection, in fact, is the triggers for local adaptation. It's really the force that allows the phenotypes that are the adaptive phenotypes to diverge from one population to the other, right? So natural selection triggers local adaptation, but not in all situations. In fact, the fact that we have normally gene flow between two populations in an entire, let's say, meta-population implies that we have an homogenizing effect between, under the genetic point of view, between the population, right? So in order to trigger local adaptation, spatial divergent selection needs to overwhelm this, let's say, homogenizing effect of gene flow. The higher the gene flow, the higher have to be the natural selection to trigger local adaptation. If this is the case, of course, what happens, in fact, is that natural selection really prompts or fosters a local diffusion of genetic variants, in fact, in one population and some in one population, some in another, that allows to native population of one particular site to present what is referred to an adaptive advantage, right? So, in fact, higher fitness to any other, with regard to any other population that comes from another site, right? So, does it mean higher fitness? Of course, this is a very basic concept from evolutionary biology. They have higher survival, right? So it confers higher survival. Local adaptation deals with survival, deals with population survival, right? And in fact, if you could move, let's say, ideally, some individuals from population 1 to population 2, they would decrease their fitness. So this is what you see here on the right in this slide. This is the typical pattern of local adaptation. So what we call normally local versus foreign criterion, right? Basically, as Stefan has already very well explained, we have come to a lot of methods during let's say our the progress of evolutionary sciences to try to understand the basis of local adaptations, finding the genomic basis of local adaptation. We have some methods in particular that are from some of them are summarized in this table on the left. Some purely population genetic based methods are based on genetic differentiation between populations. So we normally look at some population differentiation in terms of FST, for instance, or other metrics that rely with the genetic distinctness or differentiation between population. We have historically QTL mapping, for instance, or genome-wise association studies. But in fact, the topic of today is more on one more or less recent approach, which is the one that Stefan introduced during his presentation, which is this gene environment association studies, or genetic environment association studies, or environmental association study. You can find all these kind of definition in the literature. They are more or less they refer more or less to the same approach. I will explain in a bit in a while the subtle differences between one another. But in fact, what we do with this genetic environment association is just to look at genome-wide association between the genome of a species and environments, right? And this particular approach, in fact, is the one that would allow us to really detect if there are spatial signatures of selection, in fact. So we are basically correlating the spatial distribution of a potential trigger of divergent selection, as we can see, for instance, here in the figure on the right, temperature, for instance, with the present or absence of a genotype or an allele frequency that has a particular position on the genome, right? So this is basically this opens this approach, of course, opens the way towards landscape genomics, which is in fact this research field that really aims to identify both the environmental factors that triggers local adaptation, right, and the gene variants that subtend or that underlie local adaptation. So the aim of landscape genomics is not only just to find some gene environment association, but also to give a biological meaning to this association, if it is possible. We want really to arrive to give a biological meaning to the genes that are associated with this particular locus on the genome that is associated with the environment. So I will go very fast here because Stefan already said basically all the information necessary. emergence of landscape genomics, of course, is around the 2000. This is because, of course, of the flourishing of this concomitant flourishing, I would say, of next generation sequencing technique and public environmental databases of more and more resolution. And of course, by the way, this flourishing of next generation sequencing, which has come to be more and more affordable, even for for researchers that studies no model species, is not only related to landscape genomics. In fact, genomics applied to conservation allowed to perform studies to estimate the population parameters that were unable, in fact, by using simple neutral markers like microsatellites, for instance. As you can see here on the right, the figure on the right, we can accurately, for instance, estimate migration rates by using genomic data. Of course, estimate local adaptation, as we are seeing now, but also outbreeding depression and so on. So landscape genomics, the framework in which landscape genomics works is much bigger than simply landscape genomics, in fact, right? It is a method that we can use to detect a specific aspect of the entire picture, which is local adaptation, which is something very important because it relates really to population viability, definitely, right? Population growth or viability. So the population also to tackle environmental change. In other words, to what we call adaptive potential or evolutionary potential. Which is the expectation of these methods? In fact, what are we able to detect by using a gene environment association approach or a landscape genomic approach? Basically, what we in this picture on the right, in fact, we can see that this idea of evolutionary potential that I was mentioning just a while ago is not something abstract. It's really something concrete. The evolutionary potential is something molecular, as a molecular basis, in fact, and this molecular basis is principally genetic. We have also some epigenetic effect, which is not very well understood until now. In any case, we will focus on the genetic one here. In this, the genetic component, in fact, of the evolutionary potential, which can be, by the way, differentiated between into two main components, which is, we have a genome-wide neutral component, we have an adaptive component. When we talk about landscape genomics, we talk about the adaptive component. We want to detect really the genes that underlie adaptation to environment, the genes that underlie local adaptation. So by using gene environment association, which basically rely association between genotypes and environment, we are normally able to tackle genes at large effect, right? There are some evidences that if we switch to allele frequency, if we use instead of genotypes allele frequency, we are a bit more able even to detect, let's say, loci with small effect on the gene. So polygenic adaptation. But in fact, polygenic adaptation is a bit harder to be detected. And this can be a problem because, in fact, we know that adaptation to environment is normally can be at least even polygenic. So, of course, if we combine these two, the idea is if you combine these two approaches, the gene environment and this environmental correlation method, which relates more to the allele frequency, we probably expand a bit our spectrum of detection of, let's say, the molecular basis of local adaptation. So in this part of the presentation, we'll go into a bit more technical detail that relates to different aspects of landscape genomics. If you want to conduct a landscape genomic study, of course, we need to start from devising a proper sampling scheme, right? So in the case of landscape genomics, since we want to detect local adaptation, a good starting point is always to, would be at least always to characterize the environmental niche of the species of interest, right? And the goal is always to sample, of course, as much environmental variance as possible, because if we are able to do that, we augment our chances of sampling adaptive variation, right? So where to sample? One approach might be the one to look at this map, for instance, and look at the areas that are occupied by the species that corresponds to the most diverging, for instance, environmental condition. Because if the species is able to inhabit both, probably we can expect that there is some adaptive variation that underlie this capacity, right? So we have to respect, in fact, this idea and this concept of environmental representativeness. So sample as much environmental variance as possible. How much sample? During let's say, after a simulation study, which was conducted by Oliver Sermoni, basically Oliver did some simulations that were able to discern which one is the most influencing factor to augment statistical power in landscape genomic analysis. It takes into account several aspects: sample size, the number of individuals, which is sample size in fact, but the position of the individuals in the study area. And in fact, it turns out that the most important factor which compose a sampling strategy or a sampling scheme in landscape genomics is sample size, is the number of individuals that we sample. It's not other aspects related to the position of the trap of the individuals, which of course by assuming of course that we apply the environmental representativeness principle, and basically he was able to identify some rule of thumb, some thresholds in different let's say demographic scenarios, and in particular if you work with species that present high gene flow, so purely structured, let's say. So the rule of thumb is normally to sample at least 400 individuals, right? In fact, if we sample less than this threshold, the false discovery rate, so the number of false discoveries in our study might be higher than 50%. So there is basically a 50% chances that we are dealing with a false positive. In the case of, in the opposite case, in the case of very structured populations, so low gene flow, in high adaptive divergence between the population, we can reduce this effort until 200 individuals, where in fact we have a false discovery rate of about 20%. So we can expect 20% of our results to be, in fact, possibly null. Let's now have a look to the required information for conducting a proper, say, landscape genomic study. Starting from the scratch, we need, of course, an environmental layer. We need environmental data, and we have two possibilities to derive this data. One possibility is just to go to the field and direct measure the environmental variables of interest, the one that we think to be involved into selection. This is, of course, probably the best approach if you could implement this.
because basically you are able to go at the micro scale to characterize really the micro scale, and possibly selection acts, in fact, even at the micro scale. But in fact, at least to my experience, the most used approach is the other one. Is in fact to go online on the web and intercept some public databases, like the one, like for instance, a very famous one is WorkClean, for instance. Now we have also another one which is called NVRM, which is providing even supplementary environmental variables. So all this data set deals with temperature, precipitation related variables, and other indexes, derived indexes of aridity, for instance. These are completely free, so you can really go online, you digit on Google WorkClean, and you will find the proper data set, and you can download the information. But take into consideration that at least to my knowledge, the maximal resolution by which you can download this information is 1 kilometer square, which can be sufficient. But of course, we are losing all the signals that are the signatures of selection that where in fact the environmental triggers of selection act at minor resolution, right? Minor scale. It is related to the question of before. One important thing that which is important in general, because very let's say correlated with this type of environmental information, is redundancy. So when you download environmental information, when you deal with environmental information, essentially you deal with the redundant information, and this can be a problem for landscape genomics in general, for all the approaches that implies an association through some statistical modeling between two variables, and so also in landscape genomics in gene association models. This causes numerical problems of convergence, computational burden, which is essentially unuseful. So the suggestion is always to download this information and deal with appropriately with the problem of collinearity, right? So the fact that one variable can explain in some way, somewhat, the variance which is contained in another variable. So the suggestion really here is to read dedicated literature and adopt techniques to deal with the collinearity and try to remove it. Of course, the second information, which is very essential, is the genomic information, right? So which kind of genotyping approach we can use in landscape genomics? Basically, we have several possibilities. We have both, in fact, unpooled approaches, where that results in precise genotypes and allele frequencies that spans from whole genome genotyping when based on SNP chips, for instance. But also we can use, and this is I'm seeing particularly used in some non-modeled species, something that could be of interest for us today, which is what it is called target genotyping. So we know we have some, let's say, we have some idea about which genes would be potentially involved into some adaptive responses, and we go exactly there on the genome, and we genotype the genes of interest. This is target genotyping. And of course, we have some complexity reduction techniques like RADSEG, which is very much used. And yes, and this is related to the unpooled approach. But of course, we have the specular pooled approaches that we can use that will result, of course, in allele frequency. So landscape genomics can be used also with pooled approaches. But of course, with the drawback that we will not be able to exactly identify from where, in fact, the spatially the signatures of selection comes from, because everything is put together, right? But in principle, we can do it. Yes, and one important thing, to me at least, when you start thinking about a landscape genomic approach for your own study, when you produce genomic data, you need really to be conscious of the fact that if you can avoid this problem of ascertainment bias, just avoid it. So include all the, if you use a SNP chip, for instance, include all the population that you are planning to use in your study to discover the SNPs. Otherwise, you would end up with some biased results, for sure, in terms of estimation of population structure or demographic, let's say, history, and you will miss some signals of selection. I was just mentioning the fact of the population structure, the study of population structure, which is something, of course, of key importance in the case of landscape genomics. If you take a landscape genomic paper, you will definitely find everything, at least one paragraph or one section of the paper that describes population structure analysis. And there is a reason why for that. And the reason why is that population structure can act as a potential confounding factor when we are trying to associate environment and genetics, right? And this is because, in fact, the output, the general output of the evolutionary output of some neutral forces like genetic drift, gene flow, genetic surfing, founder effects, can really result in, let's say, a spatial arrangement of the variance across the landscape, which can be essentially very similar between neutral markers and markers under selection. If we imagine, let's say, from the past to the present here, let's say the evolution, the spatial evolution of a neutral locus in this picture on the left, and of an adaptive locus following, for instance, a geographic expansion, right? We can see that in some particular condition, and especially when we have an environmental gradient that follows the direction of the direction of the expansion range, we can end up with very similar spatial distribution between the neutral locus and the adaptive locus. So this means that if we apply a landscape genomic approach, if we do our gene environment association study, the probability, in fact, of detecting a significant signal is in fact increasing in this situation, right? So we have to take into account the demographic history of a population. This is the take-home message of this slide, in fact. And there have been some interesting studies about this topic, and one which is to me something important to take into account is this one. In fact, this is a study from Frischot and colleagues, quite recent, and a simulation. This is a simulation-based study where basically they simulated in several scenarios some an expansion range, let's say a geographical expansion of a population into following a given axis of expansion, and then they carried out gene environment association analysis by moving, let's say, the main axis of the main direction of the ecological gradient, right? And they tested how much false discoveries they have for each combination of direction of expansion and direction of the environmental gradient. And the result was quite surprising, in fact, because the scenario in which we are able to minimize the false discovery rate is in fact the one when we have an alignment or something which is very proximal to an alignment between the main expansion axis, the main direction in which individuals move on the landscape, and the direction of the ecological variation. Of course, this is something counter to intuition, but in fact it can be also an opportunity for those of us that are studying spatially expanding populations, because in fact in these cases a landscape genomics approach will be apparently very appropriate to detect spatially divergent selection, right? And in fact, the worst situation in every case is when we have like a 90 degrees difference between one expansion when these axes are perpendicular. This is the worst case in absolute. So if you want to apply your landscape genomic study in your specific case, just think also probably at the biogeographical history of your species, right? Because you can find probably some interesting expectation from this work. So how can we use then population structure analysis practically in our landscape genomics approach? So basically what we normally do is just to quantify some numerically. We need to quantify numerically, in fact, population strata. What we need to normally we define that this quantification like population structure variables, right? We need to derive population structure variables to be introduced into the gene environment association models in order to correct for the potential confounding of the demographic history. So basically, we conduct some. We can use some very widely used approaches to detect and to estimate population structures. Both model-free approaches and model-based approaches, but let's say in all the cases, the thing is just to quantify this. Let's say some way the evolutionary history, the demographic history of a species, but also to define how many population strata to use in our models. And to do that, we need to quantify probably the very famous key parameter, which is of course a simplification of reality in the vast majority of the cases. But we need that to, in fact, to, let's say, quantify some way the effect of population, the potential effect of population structure. So we can do that with a structure-like analysis, for instance, as we can see here on the right, or also with multivariate analysis like PCA or DAPC. And in fact, we have a lot of possibilities here that we can use to quantify population structure in landscape genomics. We have some spatially explicit methods, for instance, like test tree that we can use, and all, let's say, the structure-like software. Of course, since we are talking about genomics, classical software like Structure starts to be a bit. They can have some let's say numerical problems. Not numerical, but computational, I would say, because it will be a never-ending analysis probably when using Structure with more than let's say 5,000 markers. But fortunately, there have been some development on this side, and then there are software that can perfectly and efficiently provide a structure-like software, like Admixture, for instance, or the fast structure version of structure. In fact, take care of also. General suggestion is to take care when you use one approach about the assumptions of the underlying model, especially if you use a model-based approach, the underlying assumption of each software. Regarding, for instance, R-defined equilibrium, linkage disequilibrium, the fact that you can use or not to use, let's say, kinship within your data set, or you have to leave kinship from your data set. There are something that we need to take into account. So once we have all the information, once we have derived our environmental information, for instance, from a public database, which I repeat, this is a bit the standard of probably, at least to my experience, of the vast majority of the landscape genomic studies. We have, of course, the genomic characterization. We have estimated population strata. Then we can finally run this gene environment association models or gene environment association analysis. Of course, just to stress again the concept that the fact that we are able to match this information derives from the fact that we are working with georeference data, right? So in fact, it's the geographical position of our individuals that allow to match properly this information, right? And this is also something that poses some concern if you are not studying SSI species, but for instance, species that are moving a lot over the landscape. Normally, the classical approach, even with these species, is the one of taking the GPS coordinate, a static GPS coordinate in some place where the species was observed, and derive the environmental conditions at this site. But of course, if it would be possible to follow the radio tracking, for instance, the animals across the landscape, evaluate the frequency by which it inhabits, or it moves in its home range, well, the environmental characterization would be much more precise. And the more precise the environmental characterization, the better it is, of course, for a landscape genomic study. So we have two main approaches to landscape genomics, univariate and multivariate models, which is not strictly in the classical statistical terms, because it refers not to the, let's say, the predictor variables, but rather to the response. Because in fact, univariate models, you will see in a while, model one locus at a time, while multivariate models are able to model and to associate an entire genotype matrix or an entire allele matrix at one time, right? So univariate and multivariate in this case is referring to the response matrix. Among the most used and probably famous univariate models, we have, for instance, the spatial analysis method, SAM, the one that Stefan developed, from which derived, for instance, Sambada. So there are softwares that are very well cited. They apply multiple logistic regression by regressing one single genotype at a time. But also we have like the famous latent factor mix models. So this is probably the more used approach, which is the one which is normally referred to as LFMM. And then we have some other approaches which are based on the population differentiation, but with an explicit consideration of the environmental characteristics at each site. This is the FST-based methods. And then we have the multivariate approaches that are coming really to be much more and more used. the most important one is probably redundancy analysis or partial redundancy analysis. Some examples from the univariate approach. As I was saying, these are approaches that test single locus relationship with single environmental predictors, right? So they are univariate in the response and univariate also in the predictor in the design metrics. Of course, the design metrics can use population structure variables, right? As we define in a while. So in this study, for instance, we applied, we used one of this software, particularly Sambada. So the software was Sambada, and we studied the potential local adaptation of these autochthonous bovine populations from Uganda. So each red point here basically is an individual, is a cattle, and we derived on the environmental side the potential distribution of a parasite, of an important parasite, which is Theileria parvia, which causes theileriosis or East Coast fever, which is really a very important disease in sub-Saharan Africa because it implies a lot of economic damage, and it is fatal in a lot of cases, but not in all cases. Because in fact, there are some populations in this part of the Africa, Uganda and Kenya, that are resistant to the disease. So the hypothesis here was to, in fact, detect the molecular basis of this resistance, the local adaptation of these populations to East Coast fever. We applied this approach. At the end, we obtained really for each site on the genome what we normally call the Manhattan plot. So the statistical significance of each locus on the genome, right? And in fact, we were able to define to detect some outliers, some of which they also had some interesting biological meaning. In fact, these outliers, in particular this one and another one, they were related to apoptosis to cellular pathways that deals with apoptosis in one case and regulation of the lymphocyte proliferation, which is something interesting in this case, really, because Theileria parva, by the way, promotes lymphocyte proliferation during his infection, right? So this is basically the way, through an example, by which landscape genomics start from environment and just arise at the gene level, right? And if we use a species with a mapped genome, with a lot of information like cattle, of course, for instance, cattle, we are really able to give a biological meaning to what we are finding. In the case of multivariate methods, what we do is, in fact, we model everything together, right? We take the entire matrix of genotypes and we associate this entire matrix with the entire matrix on the environmental variables. So the philosophy is completely different. And what we aim to find is just these sets of loci, orthogonal sets of loci, so independent sets of loci that covary with independent sets of environmental variables. So we obtain normally a representation like the one we can see here. This is a study conducted on a conifer species from North America, Pinus contorta. And here we can see basically how each locus, in this case it is a genotype, it disposes in the RDA space. So loci that are nearby in this space are loci that are similar. Let's say that have a similar that are correlated with the similar environmental variables, right? So we really see like cloud of loci that moves together as a function of a particular environmental pressure, right? So the black arrows here are the vector of the environmental pressures, and each point, in fact, is a locus on the genome. And we can find a group of outliers that moves together following this particular environmental condition. And of course, we have we can plot. If we have a map genomes, we can obtain exactly as before, as here, a Manhattan plot. Also in this case. How does it work? This redundancy analysis. There are several steps that we can use, and basically we reduce information to we have two approaches. One, simple redundancy analysis, and partial redundancy analysis. Partial redundancy analysis allow us to use also population structure. So it is the recommended approach, in fact, in the case of landscape genomics, because we are able to really introduce into the model population structure. What redundancy analysis does is just to reduce information to the supposedly adaptive variance of your data set. So basically, we are modeling adaptive variation as a function of environment. And then we obtain this linear combination of this RDA ordination axis, the ones that you can see here in the small figure at the bottom of the slide, RDA1, RDA2, RDA3. There are in fact combinations or linear combination of the environmental variables, so that the individuals or locus, depending on which what we want to display, they really dispose in as a function of the, let's say, the environmental stressors, right? So one interesting output from RDA is the possibility of really displaying in one single plot environmental information, the genetic information, and also the individuals, of course. So this RDA3 plot combines, let's say, the representation of individuals, that is color point on the graph, and of course they are disposed based on their similarity. So, for instance, the yellow and light blue point here, they are genetically similar, of course, and they are in fact positioned in the same corner of the plot. This means that probably they have a similar genetic composition. And of course, we have these arrows that are also the, let's say, the environmental stressors. So we can reconstruct which are the environmental stressors that pushes some individuals in some parts of the graph, and some other in the other. And then they have this cloud of points in the central part of the graph, at the origin of the graph, which are basically the SNPs loading, right? So we can also have a graphical idea if there are some SNPs that are those that are responsible of the specific position of the individuals, right? So we can in fact in one single analysis obtain a lot of information, which is not possible to obtain with the other approach. So if we compare these methods, what do we find? So that based on some simulation studies conducted based on different demographic models, expansions models, isolation by distance models, or island model, the authors were able to compare the performance of multivariate methods on the x-axis, RDA, it was redundancy analysis, and the performance of one univariate method, LFMM, for instance. And we can see that in some cases, and they also used different sampling strategies. This is the reason why we have repeated say, repeated measures, if you want, for each demographic model. And we can see that, for instance, for some models, the methods perform pretty the same, but generally RDA outperforms LFMM, especially in the isolation by distance model, where RDA was always the power from RDA was always perfect one, while we have a lot of variance depending on the sampling scheme used in the case of LFMM. So we can say that sampling scheme, of course, affects some way the performance of the meter. Is not something that we don't have to take into account if we want to make our landscape genomic study. Both meters present similar performances in some cases, but generally the RDA performs better. It has higher power, especially in the case of isolation by distance. If we refine a bit this analysis and we introduce also the potential impact of the strength of selection, and we look at the power of the analysis, we see that so in this representation we have on each rows different demographic models as the one we have just seen the other picture, and we compare the performances of multivariate against univariate model. We see that in general, when we have strong selection at the top of the graph, the power is always very good. It's good in both the cases, both in univariate and multivariate. But the more, let's say, the selection become weak, the worse the univariate model starts to perform, while the multivariate model performs a bit better. So what we can say is that again, multivariate model seems to be a bit seems to perform a bit better than univariate one in a vast combination of demographic and scenarios and sampling schemes. But importantly, and this is a very important point to me, is that RDA seems to be more able to catch or to detect weak selection, which is probably the one which is involved into multi-locus selection, so a polygenic adaptation, which is of course something very important in our when we study local adaptation, because a lot of in several examples we know that in fact adaptation to climate, for instance, in several species is due to polygenic adaptation. We have no one major gene. This is also
sorry Elia, because we are already 15 minutes late, and otherwise people will blame you because they will not have coffee.
Yeah, sure. Okay. So I will finish in a while. So basically, we have also better power, and when using multivariate approaches with regards to when compared to univariate one. And then the very last part of this presentation is about the application of landscape genomics in conservation. So one approach which is very recent and is the one that have to do with the calculation of these adaptive landscapes. When we have detected our association with the environment, we can really calculate something which is called an adaptive index, and calculate which is the genetic similarity under an adaptive point of view across the landscape. So in this map, for instance, similar color correspond to populations that in fact present a similar adaptive makeup, right? And if we are able to calculate that, and we consider that the climate is changing, in fact, so the same place on the landscape will change their environmental condition. We are, of course, by intuition, we can understand that natural population are going to be maladapted. So we are going to disrupt, in fact, the local fitness optimum that evolved in the last century of years, right? So we have this concept of maladaptation or adaptive lag, and a way to calculate this adaptive lag is to just project this adaptive index that we calculate based on the results of landscape genomic and transpose it to the future, and calculate the difference between the present and the future to estimate what we define normally genomic offset, right? So we can present the population, we can identify the area on the geographic range of a species that are more exposed to climate change under a genetic point of view, right? And this, of course, has great potential implication for conservation. So just to finish, some few take-home messages. So that, of course, identifying the basis, the molecular basis of local adaptation is central for conservation, as we can I've already said, just said. And landscape genomics is really a powerful approach to detect patterns of local adaptation across the landscape. Under a statistical point of view, combining multivariate statistical approaches and univariate statistical approaches is probably the best approach, because this allow us to decrease false discovery rate and increase true detections, so the power of the analysis. And one suggestion is also to, if possible, to combine analysis by using both genotype and allele frequencies, just to enlarge, of course, the spectrum of detection from major to small effect loci, which are expected to be involved into local adaptation as well. And then we have just seen in the very last part of the presentation some potential application for conservation, this idea of adaptive landscape and genomic offset. And then to finish, one very final remark is regarding the problem of validating landscape genomic results biologically. I mean, so give we obtain an association, but in fact we don't know if this association is a false positive as a matter of fact or not. So approaches that can be used relies to validate this are experimental approaches basically. We can apply reciprocal transplants, common organelle experiments, for instance. We can verify phenotypic effects through genetic transformation, or even verify the effect of a candidate loci through gene editing, for instance, or putative casual sites as well. So I just finished 20 minutes. Yeah, I thank you very much and for your attention.
Thank you. It was a very comprehensive presentation of these approaches, very interesting. So now we introduce the first speaker of the second session.
Okay, so the speaker for this session, the first one is Mario Barbato, and he's going to present us some interesting results about application of landscape genomics to livestock species. So Mario, the floor is yours.
Okay. Everything is settled. So I'm older than I look. I will just sit down. So first of all, I really need to thank Elia. Well, certainly for the thorough excursus that it provided on the topics of the tools for landscape genomics, but also because it basically removed sort of half of my presentation by explaining stuff way better than I would have done. So thank you, Elia. Meaning we will be in time for, the social dinner. All right. So another thing that I'm quite pleased with is that I will have, it's going to need very little effort to convince you that exploring this kind of topics is worth. And I will start reminding you, although it's not needed, that the climate is changing. So not very novel as an information, but bear with me. And let's say the most optimistic provision say that there will be an increase in 2 degrees, and I'm referring just to Europe now, in the next 80 years, whereas the, let's say, less optimistic are suggesting something like a 4 degrees increase in the next 80 years. And of course, that means troubles right? Because with temperature, then you will have changes in precipitations, and it's going to be, well, let's say, a very unpleasant situations. With extreme environments will become even more extreme. At the same time, human population is growing. Some would say at an alarming rate, but let's just stick to is growing. And that means, or at least the prevision said, that there will be a correspondent increased food demand. And specifically, something in the order of a double amount of livestock products, to make an example, by 2050. So I'm not talking about 1 century from today. I'm talking about 30 years, less than 30 years for today. Now, it's very clear that there is a collision here. So we have an likely increase in animal products, which will have to deal with a climate which will change and will affect animal production, and will affect it, well, decreasing the productivity, the overall productivity, right? And here I've listed a couple of the aspects that might be affected. And it is for these very reasons that we need to think thoroughly of how to tackle the issue. And I want to be very clear, it's something that we cannot just do with one single tool. It's something that needs multifaceted approaches. All right, so there's going to be an increase in diseases. Well, it's going to be people that study diseases that work on that. And competition for natural resources, we require a different, let's say, expertise. For these reasons, a project like the IMAGE project came to life. And it is a project that attempts, of course, we do our best, let's put it this way, to try and tackle the issue using, from different angles and including a lot of different expertises in the process. It is a Horizon 2020 project, which includes multiple partners for all Europe and even non-European partners. And as I mentioned, it has several, let's say, aims that we'd like to tackle. And as a group of researchers, of course, we are, let's say, we can offer our efforts towards what implies genetics and genomics, and that's the bit that as a group and colleagues, we are trying to understand, to better understand and provide possibly some answers and also some idea on how to deal with the future scenario. And specifically, so I'll just read it, is enhancing the usefulness of genetic collections to allow the livestock sector to respond to new environmental constraints and market needs. Meaning we need to try and make the genetic resources as valuable as they could be. Now, well, we are in an agricultural faculty, so it shouldn't really surprise you that we pointed at one of the nicest and fanciest species on earth, which is sheep. But true reason, although, well, first reason might be because they are fluffy and they're nice. But second is because as many as small ruminants in general, you have a situation in which you have very conserved small local breeds all around. Well, it is a a breed that inhabits basically any possible environments, even those environments which are not really likable at all, and yet they are there. And the reason is because they tend to be very rustic. However, of course, there are many breeds which are cosmopolitan, which are highly selected, and those are probably the most famous, those that we think about when we think about sheep. But the real richness, at least in the context of what I'm showing you here, the real richness is within the many local sheep breeds that inhabits any continent, really. including Europe. And of course, all this diversity exists, well, for all the population genetics reasons that you might think of, but also, as suggested by the previous presentations, because they ended up in environments which are diverse, and through time they adapted to this new environment. So as my boss likes to say, it's a very nice natural experiment ongoing, right? So to investigate this aspect, so we defined, we wanted basically to identify which were the genomic components which changed in frequency basically to adapt to all these different environments. So basically, this nice figure that I stole from somewhere. Well, this is the bit that we are more interested in, right? Genotyping environments. We want to perform environmental association analysis. And please refer to Elia's talk for all the details on how to do that very nicely. What I'll do now is just to go through the steps that we went through to try and apply the method, the theory, in a practical case. So first thing was, of course, to sample stuff, right? And when we're talking about landscape genomics, we need to sample basically two things. Well, we sample animals, all right, but we want to sample the environment. And of course, I now remember that Leah was mentioning, well, we actually know that of all the different aspects that you should take care of when you are designing a landscape genomic experiment, be sure that you have enough animals, all right? It was like over 400 animals allows you for a very nice FDR. And yet, a second later, he also said, well, that of course is important as long as you already sampled, you have a representativeness of the environment. So we had no issues in terms of a number of animals. As you can see, we ended up with more than 1,000 and almost 1,500 individuals. What we had to make sure was to sample the environment properly. And to do that, we relied on Oliver's expertise, which basically, and I think, is it tomorrow you will, yeah, so tomorrow we will explain in detail, the process, the rationale behind that. But basically divided the sampling area in homogeneous cluster of let's say climatic variable. Allow me to be a bit loose with this. And then we made sure to have enough sheep per cluster so that we were basically having a thorough representation of the environment of the sampling area. Long story short, we ended up with 82 local sheep breeds. Again, a bit less than 1,500 individuals. We used high density DNA arrays, so SNP chip, which luckily exist for sheep. And at the same time, we collected bioclimatic variables, plus, well, in this case, I just wrote elevation, but we actually used this 19 bioclimatic variables to also generate other synthetic variables. So we do have the genotypes, we do have, the environmental information. And now we go to, basically putting them within a tool that will do the analysis, all right? And we chose to use a couple of approaches. The first one is, well, have been just mentioned, is LFMM, which is again a univariate approach, meaning that you will test one single environmental variable against, let's say, as a predictor to explain one I think, alleles with LFMM, yeah, or genotype, well, one single locus, that's for sure. And then you just, well, the software will repeat the analysis as many times as necessary to cover all the SNPs, and you will do it for all the environmental variables you are interested in. Well, it's not an issue, but let's say a feature, let's put it this way, of LFMM is that it corrects or attempts to correct for a population structure, and Elia was very highlighted how important it is to correct for population structure, unless you want to get a false positive, as if it was raining. So it does it by itself, but not totally. We need to provide some prior information, and this information is a general K value, which means in how many populations, subpopulation we think we should divide our data set. So keep that in mind. Second approach which we tested, well, we did not just tested it, we applied, is the redundancy analysis, which is, again, it starts to be quite present in recent research. And it's very, very interesting approach for the reasons which have been already exposed. And main thing is that it allows you to test, in a multiple regression scenario, it allows you to test regression of multiple response variables, meaning that I'm not testing a single SNP each time. I'm testing the environmental variables against the genome. And that's, of course, very interesting because, and I will make, again, I don't need to add much to what has been already said. What I really like is that it is, to my point of view, closer, to the real complexity of nature. We are still doing models, so they're going to be wrong nonetheless, but this is maybe not the worst, all right? Now, yeah, I will just skip this one because you already seen this image. I will just point out the nice title they used for the relevant paper, right? And it is a paper which I absolutely recommend. So I will just pretend that we just had two issues here while doing the analysis. And issue number one is that, as I mentioned before, LFMM needs this K parameter to correct for population structure, all right? Thing is, how many Ks, which is the right number to describe, the stratification in our population. Second issue is that RDA, as a first step, starts with a multiple linear regression, meaning that, we have to take care of multicollinearity among predictors. We cannot just have as many predictors as we want if they are telling the same story for the reasons that have been already mentioned. So issue number one, well, a very common thing is to perform a structure, a population, more in general, a population structure analysis. In this case, we use SNMF, if I'm not wrong, and that's what you get. Now, normally, let's say general approaches would tell you, well, now it's very easy. You just need to take the lowest K value in your analysis. Truth is that we believe that, pointing at a single K, it's substantially, you will never get a right K. That's the point. There's no such thing as a right K, especially when you have a very many populations, meaning subpopulation in your data sets, which will have substructures themselves. So it is very complicated. The reality certainly is not model-based. That's the point. So if we wanted to, let's say, go with the most common approaches, well, one suggestion would be, well, just take the K value where you have the, the elbow of the curve, if you want, or maybe again, just take the smallest value in your analysis, or why not, you know, whatever point in the middle. I mean, they are not, none of this is exactly correct as a solution, and none of that is perfectly wrong. They are, if you want, all right. No more than that, though. And for these reasons, we attempted to do something, which, of course, I want to disclaim, is not going to solve the issue, not at all. It was just our approach to try and let the data speak, basically. You will evaluate if it makes sense or not. What we did was, first of all, let me point out, do you know, can you see how wiggly the data points are, right? They are super wiggly. And that's not because those are real value, exactly. It's just because, you know, it's especially the starting point of this analysis is very stochastic. So, you know, I repeat the analysis 100 times and I will get more or less different values all the time. All right, the topology is going to be the same. The actual values will differ a bit. So what we did was, as you can maybe imagine, we just repeated the analysis many times, I think 100 here. And we just drew the distributions of the values for each k. As you can see, those in pink, those are violin plots. And what we did was just to then consider the median of this distribution to draw a line. And I'd say it looks not as wiggly as before. This is the starting point. Second thing, well, this of course makes a nicer plot in a way, but it's not solving the issue, is it? So what we did was to, you know, I just spent some time pointing my finger against old school methods. So we applied an old school method. It's very similar to if you want to an Vanno approach to structural analysis. So what we did was to compute the first derivative and then take care of the delta between the points of the derivative. So the distance, the informativeness of each k, and we just chose the key value at which the informativeness, if you want, started to be basically negligible or close to negligible. Again, is this the solution to the critical and highly discussed best k problem? Not at all. Nonetheless, to apply LFMM, you need to provide a k value. And that was our attempt to make it less, if you want, less arbitrary. Yeah, and I also included this very low resolution figure for some reasons, right? Let's pretend it was an aesthetic choice, all right? So it's like a 90s vibe, you know, 8-bit. So second issue, that was to remove collinearity, so to reduce the amount of environmental predictors, right? And again, Elia discussed that a bit. And there are different approaches. One that might actually make a lot of sense would be to just, you know, run a PCA. You get synthetic variables, which will be, of course, orthogonal. So no multicollinearity at all. Problem is, well, problem will come like exactly at the point when you want to try and interpret the results, because you will have synthetic variables which will contain loadings from different variables, and it's not a given that it will be all coincide in what you expect it to be. So it can be challenging. And well, to the best of my knowledge, I don't know if there's a solution for that. I don't know. What we chose to do, though, as another approach, was basically to iteratively remove variables using the BIF, of course, until the BIF went below a specific threshold, which we find to be 2.5. It's not that it's not an arbitrary value that we chose. We basically scanned the literature as much as we could to understand which was a reasonable value for VIF to, let's say, be safe enough. But let's remind that, as perfectly explained before, with environmental variables, it's, well, I don't know if I can use absolute sentences here, but it's very hard to have completely uncorrelated or independent variables, okay? So we're just trying to do our best. So long story short, eventually we ended up we started with 20 and more variable. We ended up with 7 environmental variables, which are listed here, right? So let's see. We had the environmental variables selected. We defined a k value. Basically, we are ready to run the analysis. We performed both analyses, and these are, let's say, the first results from the partial RDA. And this is the famous tree plot, which has been discussed before. So the yellow dots are individuals, and the grayish, dark gray dots are the loci, and you see dashed blue lines. I'm sorry, because I left the not very explanatory labels for the environmental variable, but these are the environmental variables, okay? So, and you can see the phenomenon that Elia was mentioning, that, you know, you have individuals which basically group together. By doing that, you know, we were able to identify which were the SNPs, which were sort of outliers in all these clouds of information. And that's the main result of the RDA. And LFMM, well, I won't make a long story with it. You know, it's a model, and you get sort of a Manhattan plot, and you select values which are significant. So again, I'll try to shrink it to the essential. Using the PRDA, we got something like 2,300 SNPs in total, and a bit less than 100 SNPs with LFMM. With 52 of these SNPs, which were in common. And actually, I was going to ask Elia for a comment if there was still a reason to be for a univariate tool like LFMM if we have the RDA. And I think that this can be, you know, a reason to still use it, to compare and to see if you get some common signal. Because as I just mentioned, with LFMM, we are sure that we will still be able to intercept whatever is very strongly selected. So if you want, it's giving us an additional layer of information on the PRDA data, if you want. From here, we just tried and understand which genes were intercepted by those SNPs. That's where we get. Well, I won't read all the numbers. What is interesting is that we still got some 52 genes which were identified by both methods, right? And now, of course, I won't start listing all the 1,200 genes. You know, it would be sort of pointless, and you would probably kick me out of here in no time. I will just go straight to the endpoint and telling you that what we're doing now is that we scanned the literature to see if among these shared genes, which clearly they do have a special role in the adaptation of all the sheep breeds which we sampled, you know, if there was some direction. And what we found is that there were many genes, and here I list some, that were involved with fat deposition. Beware that the trait name normally refers to how other research defined that trait, meaning that if I'm studying fat deposition, maybe due to the tail type in sheep, I don't know if you know that you have sheep which have thin tails and other which have fat tails, and that's a very let's say not infrequent kind of research that is done to find the difference between the two. And whatever they found, they say, well, this is tail type fat deposition. So I'm just referring to the trait as it's normally mentioned. Similarly, for feed efficiency and body mass and adaptation to climate, which, if you want it, I cannot make it more general than the adaptation to climate. And yet, this is very interesting. because from our perspective, what we see here are all mechanisms which are somehow involved with energy management, because, well, that's what FAT does. Well, there's also, protection from the environment if you want, but that's the main thing. Feed efficiency, okay, same thing. You have the efficiency in generating energy from maybe scarce pastures or harsh conditions. And well, I won't comment on the adaptation to climate because it's too general. One really needs to try and dig in the details. From there, then, we tried to move a little step forward, and we did geo times enrichment analysis. And we did it for shared genes and only those genes identified by the RDA and only those genes identified by the LFMM. We got no information, well, no results for the LFMM. We got results for the genes shared and the LFMM and the genes identified by LFMM. And yeah, this little cow here just trying to remind me that for these specific analysis, I had to rely on Catalan notation. Because this kind of analysis, they work very well if you have a detailed annotation. And for sheep, we are not there. We are far from there. Cattle is a bit better. Someone would use human, but then if you change too much, if you're too far away from your species, then you are including more bias. So in this case, I used cattle. And well, at first, these terms were not exactly self-explanatory to me. So as we always do, we dug in the literature, and it turns out that serine peptidases, as well as macroautophagy, they are highly involved with innate and adaptive immunity and inflammatory processes. All right, and they were associated with precipitation seasonality and annual precipitation. So precipitations, let me shrink it to the essential. Similarly, for supramolecular assembly of small molecules, which when I read it, I thought, okay, I never read, I have not a single clue of what it might be, but seems to be a very strong mechanism to inhibit pathogenic cells. And again, associated with precipitations. So to try and wrap it up, we found several strong, I remind you, identified by both methods, candidate genes, which have been already mentioned for stuff that has a lot to do with adaptation to climate, especially with energy. And we did it now explicitly, but that's the main difference. What we are providing with this piece of research is that we are explicitly saying that the environmental variable are drivers for those changes. And then again, it's that the energy management seems to be indeed the main, or among the main drivers. Also, thanks to the geo analysis, we have a strong position to suspect that also, well, not that surprisingly, fending off pathogens and infection might be helpful. And this is not unexpected, and it's not unexpected that it was linked to precipitations, because remember that small ruminants, especially, when you have very humid environment, they suffer from a lot of diseases. The most famous one might be foot rot, but there's plenty of pathogens and other vectors that thrive in very humid environments. So it makes quite a lot of sense. Are we done? Of course, we are never done. What we certainly need to do now is to try and dig a bit more on what is less explicit, if you want, on our results, and also do what Elia was mentioning at the very end of his presentation, that is to try and compute this genetic offset, so that we can also try and predict, which is the challenge, that these local breeds will face with the changing climate, and that will be a very good result, because it will tell us, the direction for real actions and to try and sort out this big mess we are in. And very last, of course, I'm just talking here, but this is a very choral work, and all these people are involved. Elia, first of all, we are working tightly on this paper, but all the other person mentioned, they helped and they are helping very much, and of course all the partners of the IMAGE project. And that's it. Thank you.
So thank you, Mario. Super interesting and very nice to see the results of IMAGE.
At last.
At last. So does the audience have one or more question about this presentation for Mario?
Hello, everyone. Thanks, Mario, for this rich presentation, this great work. My question is about one environmental effect that seems to play great role in domestic animals is husbandry practices.
Oh, yeah.
And we know that there is a great difference, especially you have a great area with 3 continents and different husbandry practices. Do you have some proxies among your environmental variables that you choose that could give an idea about this environmental effect? And how do you consider this as a confounding effect also? Maybe?
So thanks, Paolo. This is a great point, because if I was talking about cattle, it wouldn't have been the same story, because cattle, they are mostly highly managed. With sheep, well, high management is becoming a thing, of course, and in some areas of the world, it's absolutely, you know, they are sheep 3.0, you know, they are more advanced than us, that's for sure. But overall, with sheep and small ruminant is not a great issue. And we thought about that, of course, because husbandry can change everything. You know, if it's raining, pouring rain like hell, well, if the farmer will just, you know, remove the water, no problem at all. That's the thing. But here we selected local breeds which have the tendency, and that's the best that I can say, of course, I cannot guarantee it, but they have the tendency to live in low management scenarios. That's the thing. So it's very interesting if there was a variable that we could have used as proxy, you know, to basically correct, if you want, our findings. And might be worth thinking about. I don't know if you have like a suggestion for that. Might be worth thinking about it. But our approach in this case was basically to try and have populations, sheep population, which were living in the same low management conditions. That's, you know, it's never enough. It's still better than not taking it into account.
Thank you, Badr. Is there any other question from the audience? I have one, Mario.
Oh, yes, please.
And I'm sure you probably already thought about it, but I was wondering. So in your analysis, you used the outlier loci and you looked for the adaptation in these loci that, in theory, in different breeds should adapt to the same environmental constraint using the same alleles. But what if adaptation is not in the outlier, but is in those that define the population structure, because the breed itself is a local adaptation to the environment? How could you detect these loci?
Well, I think you just lose it very simply, because you know, because you cannot distinguish it from population structure. And most of these approaches that we use, they really try to do something similar, to do something that looks like, okay, I try to understand how populations or taxa or whatever would differentiate, if nothing else was going on, you know, just pure drift, you know, just geographic distance. And then whatever is outside this divergence, it might be due to something else. And that's the thing. But if the adaptive SNP lies within the cloud of, I think it's lost. But I'm happy to you know, to be wrong. I would be super happy to be wrong. And just to make, cherry on the pie, truth is that most of the adaptation works on polygenic effects, which are minimal, and is the additive effect of the minimal contribution of each selection that is really playing the adaptation game. And we are totally unprepared to get there. Although the RDA maybe is the tool that is going exactly in that direction, because it looks for interaction. Okay.
Thank you. I was just thinking, I'd look at those set of SNPs that don't overlap between LFMM and RDA, but just looking at the RDA signatures. Sure. This great amount of SNPs that, in fact, are significant in RDA.
It was actually very interesting that with RDA, of course, we got so
it is in line with the expectation, I think. And the second thing about what you, Oliver, was saying, probably with the gene environment association analysis, you are not able to do that. But probably using other approaches like population differentiation methods, like PC-adapt, for instance, which is you can take some hints about this type of adaptation, meaning really these are methods basically that works with population structure. So yeah, this can complement, in fact, gene environment association analysis.
Thank you.
So thank you very much. If there is no further question, we can move to the next speaker. So thank you, Mario. Now it's time for Badr. So we remain in the same domain of genomic solution for livestock species, and looking forward to hear what Badr has to tell us. So Badr, the floor is yours.
Thanks, Oliver, for this introduction. As you said, I will be in the same topic, almost with some difference in the question, in the scientific question to answer. So I will also, thanks Mario, because he introduced well the methods and so on. So I will not describe deeply the methods, but I will go directly to the results, to the biological aspects and so on. So, and thanks Mario also, because he introduced the environmental changes that we are living now in this era. And I will start with these basics about the impact of the environmental changes on every organism. So we have only 4 possible reactions or answers from the organism to these environmental changes, or to the environmental pressures, exactly. We have first local extinction, if any other solution is possible. The second thing is migration, where the environmental conditions are suitable to this, and if good conditions happen close to the location. The 2 other things is first acclimatization, which is phenotypic plasticity that happen through epigenetics, for example, and other mechanisms. But we have basically the same genomic material that allow to face environmental conditions differently. And the last one is local adaptation, and this is the main thing, because local adaptation is defined as genetic changes or genetic variation that allow to increase fitness, which is survival and reproduction, for sure, both, of a population in a given environment. We know evolutionary processes and so on. Local adaptation depends mainly on selection, because you have it happens when you have an environmental pressure and you select some alleles that allow to face these environmental pressures, but also depend, as shown by colleagues earlier, It depends also on other evolutionary processes like migration that interact with selection to allow or to reduce local adaptation. There are genetic drift, mutation, and also phenotypic plasticity. So it's very interesting to study local adaptation. But from this figure, we can see that it's very complicated, not easy, really. There are adaptations, examples of adaptations that are simple, like we know, for example, we have major genes that affect a trait, like lactose digestion. It's linked to a single gene, single SNP, so it's presence, absence. But there are many. The general case is we have complex adaptation that depends on many genes. Or I give this example so because it's interesting. We have here mouse in Nebraska that are adapted to the environment, adapted to prevent predators to eat them. They changed their color, but not only the color. There is the color, the shape of the tail, and so on. And genomic basis of this change of trait are complicated. They are on the same gene, but we have different locus, different SNPs inside the gene, and each locus give a different trait or affect 3 different traits in the same time. So I show this example, but there are in the literature many other examples that shows the complexity of local adaptation. So this drove us to think, because when we look at the literature, papers show that we have a gene found in a species, in the other species we found the same gene. So we thought that it was a bias, maybe a bias in the literature. I gave here another example for altitude, adaptation to altitude in humans and dogs, and then in sheep. We have almost the same gene, that is the gene EPAS one. But we thought that maybe we have other scenario. We may have different genes that affect the same adaptive traits that give the adaptation under an environment. And we can also, because we found in the literature some examples that are associated, for example, here we found in a previous paper that we have goats that get adapted to head stress differently. The first population, sweet, for example, and the other one, paint, so like dogs. So we imagined that there are many different, that 2 different traits could allow the adaptation to the same environment. So here we have a model, good models, livestock, sheep and goats, that are domestic species that have diverged recently at an evolutionary scale, 5 to 6 million years. They get domesticated in the same area, in the same era also, and they are almost raised under the same environment. For sure, there are many differences, but as 2 species, they are close. So, and we had Moroccan sheep and goats that are located in Morocco. Morocco is an extreme of many migration routes following the domestication events for sheep and goats. We have Mediterranean routes and the North African route, and also livestock came from the south and so on. So it's at the extreme. And also Morocco has a very diversity in landscape, has a very diversity in the environment. We have area where there is rains, we have desert area and so on. And Moroccan sheep and goats, this is the last thing and the most important. Sheep and goats are managed really traditionally with low input system, and they are really faced to the environment directly in general. So we thought, and this is the idea of Pierre Taberlet presented by Stefan earlier, we thought that landscape genomics, with the revolution of genomics also, if it can answer to the question of, first, if we can test the adaptive landscape concept. Stefan talk about this right, how he in 1943 maybe, or maybe earlier, he described how allele frequency could vary throughout the environment, and when we put two traits or two genes, we can have a shape differently, and so we were interested to test this aspect. And also, once we have these two close species, if we have some repetitivity in local and in the basis of local adaptation in these two close species, if we have the same genes or the same traits and so on. So for this is map of the northern part of Morocco, which is a huge area, 50,000 square kilometers. And this part was divided into cells, regular cells, 50 kilometers across 50 kilometer, and we had a huge sampling of about 3,000 sheep, local sheep and goats over all this area. And Stefan and his team sampled over these thousands of animals, 160 sheep and 160 goats to represent the maximum of variability of environmental variable, mainly climatic and also slope, altitude, and so on. So, and then we choose these animals, whole genome sequence were produced for this number of animals. And after many processing of data, colleagues have selected more than 70 environmental variables from altitude, slope, solar radiation, temperature, and rainfall, and their variation. And we did a correlation between these 70 environmental variables, and we exclude variables that were highly correlated at more than 0.8 as an R-square. And then we applied different methods. It was difficult. It was very hard at the beginning because genome information, genome data were produced at a time where we had to set up everything, and very few projects like this happened. So, and this amount of data came at this time. So we had to do many setups and so on. So finally, we chose two different selection signatures methods. First, Sambada, presented earlier by Stefan, which is a landscape genomics approach. And also we added another method, which is actually population-based method. Simple FST combined by XPCLR, but how to do it? We made a ranking of individuals according to the environmental, or the level of and the value of the environmental variable that happen when where they live, where they were sampled. And after doing this ranking of 160 individuals for sheep and 160 individuals for goats, we choose the 40 individuals, in fact, 20 individuals at the top of the gradient of each environmental variable, and 20 individuals at the bottom of the environmental gradient. Do you see? It's okay. And we compared them, we applied selection signature based on population genomics models, as I said, XPCLR and FST, to try to be more conservative, to try to look at, to verify and to validate some signatures of selection that we defined. So we managed to have we corrected different parameters and so on, and finally we end up with many selection signature. Here we have, for example, selection signature for temperature in sheep, but overall we found more than 15,000 SNPs or indel or short indels over the genomes of sheep and goats. And we've identified those variants or those SNPs comprised in 637 genomic regions over their genome, and this associated to 430 genes. We did the term enrichment analysis to find the biological processes that were enriched by those genes that were identified. Here I gave an example, just an example, but there are many, many examples. We show here the biological processes associated to the adaptation to altitude in goats. We found here a process linked to Clara cell differentiation. Clara cell are cells that are in the lungs, and they play a capital role in the respiration and so on, and it makes sense. And also we found that this differentiation of those cells is associated with high altitude in llama, previously reported, and also associated to hypoxia in rabbits. So it makes sense for this. And we found, for example, here I showed these cardiac processes that were highlighted for altitude in goats, and they already get associated with hypoxia in some other mammal species, and it makes sense. So to see, so as I said before, we were interested to show the variation of allele frequency over the gradients over the environmental gradients, to show empirically this, so to validate these theoretical approaches. So as I said earlier, we classified individuals, we rank individuals over the gradients. Here, for example, we have altitude, we ranked the 160 sheep over the gradient of altitude. And we made groups of 20 individuals, and we move by 10 individuals to estimate allele frequency, just allele frequency in outlier variants that we detected. And it was very interesting because we show empirically the variation of allele frequency in the outlier genes over the environmental gradient. We show it clearly. We did this for all outlier variants, and we end up with this figure that shows different shape of these variations of allele frequency over the environmental gradients. Here we have the outliers for rainfall seasonality. For example, in sheep and goats, you have many shapes, but yes, we have linear variation, and the variation abrupt at the end of the gradient or at the beginning, and so on. So also to validate the results, we took some example for some outlier SNPs that we identified. For example, here we have a Manhattan plot that shows the outlier windows or SNPs in sheep. And we took the outlier SNP, the SNP that has the highest XPCLR score here in this peak. Here we have an illustration. We have a heat map of temperature in July. And here for that SNP, the variation, the distribution of genotypes, we have homozygote CC, heterozygote TC, and homozygote TT in white, and we show clearly that there is a distribution according to the variation of this temperature. In this C graphic, we have the variation of allele frequency here with this blue line that show the variation of this this coincidence with the mean temperature in July. So this is from one side, and from the other side, we, as I said before, we were interested to look if there is the same signatures of selection that happen in sheep and goats. And for this, we mapped the reference genome for sheep onto the goat genome and vice versa to see if there are the same things. And over the 15,000 variants, we did not find any variant that was outlier in both species. For genes, we identified 6 among the 430 genes, but they were in different environmental associated to different environmental variables. Only one, which is CA2, was associated to the sunshine duration in sheep and goats. And similarly, we identified no biological process that was reached in both species. So we had a good example. I want to focus on it because for the adaptation to altitude in sheep, we identified a gene which is MCM3 that was reported or previously detected to regulate the expression of the gene EPAS1 that was identified in human and also other species in the Tibet for altitude. So this example, I used it to be back to the figure that I showed at the beginning. So from the results, at the inverse of the hypothesis and what was reported in the literature, we think that adaptation is not repetitive from our results. So because no genes were identified in both species, especially in this case of sheep and goats in Morocco. So we think that generally adaptation could happen through different traits and different genes. And yes, this what we expect. But here I showed the case of the gene that I showed in the previous slide, the gene MCM3 that regulate the gene EPAS1 that could affect, because this gene was associated to hemoglobin production in the blood, and it's associated to the oxygen, and it makes sense for altitude. And also we found a single case for the gene CA2 associated to the adaptation to sunshine duration in sheep and goats, but it's actually a minority or a minor part of what we identified. So at the end, I don't know, it's okay? Okay, at the end, we can say that because I want to be back to the first problematic, which is environmental changes. So we have to think, because livestock is very interesting for human population, we have to think about how to valorize this adaptive diversity. For sure, colleagues previously told about this, how to go further to identify this adaptive diversity. To maybe we can imagine in maybe in a few years we can try to integrate this in assisted selection schemes or genomic selection schemes, and to increase the productivity under harsh environment, which we expect to be more predominant in the coming years. But one thing that could be interesting from now is to associate this adaptive diversity that is detected through whole genome sequence data and so on, to associate it to neutral diversity, to set conservation priorities, for example, because there are some good indices that were theoretically developed that could combine between adaptive diversity with neutral diversity to manage these livestock species for the sustainability of the production of livestock generally. And this could be a good thing. Thanks for your attention.
So thank you, Badr. Super interesting, very nice results. It's a data set that is amazing in Morocco. I don't know how they managed to sample all around the country. And, it's really a great data set, I think.
Yeah.
So I don't know if we have the first question from the audience. So if anyone wants to ask something to Badr. Otherwise, I can start with one question. So Morocco is the perfect playground for this kind of studies because we have such contrasting condition. And as you said, this livestock is managed in a traditional way. So they really live on the field. How do you think this kind of approach could be applicable for more domesticated species? I'm thinking, for example, to cattle in Europe. Do you think that landscape genomics has a room for this kind of studies in local adaptation?
So it's a complex question, but from my feeling, local adaptation needs time to take place. So it's change in a population, not in a single individual. And it happens through generations. And the advantage of sheep and goats in Morocco, so they arrive at very a long time, since a long time, and they were managed for centuries like this. They got time to get adapted. But currently in Europe, for example, you have artificial insemination everywhere. I don't know how to. Yes, I don't know how many generation cattle are present in a location, because yes, we took the location as a proxy for the adaptation. One thing could correct, in my opinion, is if we can, with the progress in the Internet of Things and the remote sensing data collection, we can study the phenomics of adaptation. And if this can be applied, I think we can associate directly the phenomics of the phenomes with the genomes. But this kind of approach, I will not agree.
Thank you. Okay, we have some questions.
As you say, Badr, it takes a long time for natural selection, to make these changes that you're observing. And you've mentioned phenomics. What about epigenetics? Have you looked at the epigenetics of adaptation, which could happen not over generations, but even in one generation?
Yeah. We run a study over epigenetics for one single environmental variable. You know, epigenetic experiments are very complex, but we ran this study with people from Grenoble, and we found no convergence between sheep and goats, but we found a few marks of epigenetics for temperature variation in Morocco. So we identified regulation differentiation between in a single gene in sheep and in another gene in goats, and basically that they are not associated to the same biological process. And the study was published in 2021. I can give you the paper if you want, so it's okay. So this is the thing that
I guess the interesting thing would be to look at the same breed, the same genetics in two very distinct that have been raised in two very distinct environments.
Yeah, it's a question or it's a comment? No, it's okay. Yeah, for sure. But yes, we did not differentiate between breeds of sheep and goats because the landscape genomics approach. One thing that was interesting in the Moroccan case that we found absolutely no population structure in both species. So we do not have any difference. FST among breeds was 0.0000 and so on, and PCA the first second main component explained less than 1% of variation. So, and this NMF and so on, K equal 1 was the most likely and so on. So absolutely no population structure. But yes, it could be good to look at the same difference between breeds if they are raised in the same conditions. I don't know. It's okay. Answer. Okay.
I think there is another question from the audience.
Hi. Thank you for your presentation. Very interesting and very surprising how much little parallel evolution there was. I was wondering, just looking at that photo, is there a difference, like at that micro scale, different use of the habitat or resource between the two species? Maybe that can explain different strategies.
Well, generally in Morocco, we have herds like this, goats here and sheep just close. But in some few cases, yes, there are some micro differences. I will say, for example, goats can eat some tree products, some tree leaves, yeah, but sheep couldn't. For example, but in very rare cases, yes, there are a few differences that could happen. For sure, as I asked Mario earlier, husbandry practices may affect, but in Morocco, generally, we have similar condition between brackets. But yes, there are a few differences. And during the sampling, we try to take only local breeds and indigenous breeds. It's okay. Thank you.
Thank you, Badr. And now we have the last speaker of the day. So it's Arianna Bionda from University of Milan. Hi, Arianna.
Thank you very much for your presentation and for the opportunity to be here. I'm going to open. Okay, thank you. Okay. Yes. And okay. So thank you for the opportunity to be here. Today I'm going to present you some application examples of the study of the geolocalization and the relation of the geolocalization of Italian small ruminant breeds with both inbreeding but also climate threats. In fact, okay, it's not going. Well, no, I won't repeat in a very long way, but the climate is changing and this is causing also some effect on the livestock. In particular, not only the temperature is increasing at an unprecedented rate, but we also witnessed the increasing frequency, for example, of extreme events like long and very intense heat and cold waves, or extreme precipitations and a change in the intensity and the distribution of precipitations, and also, for example, an increasing number of events of droughts, floods, and wildfire. And for example, just to give you an example of what we are living, this was the situation in Europe just a couple of months ago. And all those who were here well know that we experienced an incredibly hot and arid summer, and these obviously had very severe consequences and negative consequences also on agriculture and on farming systems. But as also the other presenters said, the climate change is affecting animals in very different and not always so straightforward ways. For example, reducing the pasture and forest crop production, availability and quality, but also changing the distribution of elements and vectors and leading to the emergence of new temperature-related illnesses and the increasing morbidity, for example, during extreme weather events. And in general, the immune system, as well as the metabolism of animals that are, for example, under heat stress, are impaired, such as they can experience depression of feed intake and conversion, for example, and these impacts negatively also production and reproduction. All of this is also causing a loss in the biodiversity, but yet at the same time, the biodiversity is considered one of the key tools that we have to increase the food production in a sustainable and adaptable way to the future climate that we will face. So why studying small ruminants? Because they, sheep and goat, were among the first species to be domesticated. And now they live practically all over the world in very different managerial and environmental conditions. And they demonstrated to be very adaptable to also harsh environments and to be able to produce and reproduce in suboptimal condition. Moreover, Italy is a reservoir of caprine and bovine biodiversity, counting over 36 goat and 44 sheep breeds that are well adapted to different environments, because Italy is also characterized by a richness in historical and environmental environment, and so we have very different farming system, for example. And it is well known that both due to artificial and natural selection, local livestock population have acquired the peculiarities that allow them to survive, produce, and reproduce in suboptimal condition, as I said before. But although their production potential cannot be for sure compared with cosmopolitan breeds that are reared in intensive farms, they demonstrated to be a great resource facing the climate change, because they have a strong link with their territory. In fact, for example, they are usually reared in extensive farms and in marginal areas that would otherwise be abandoned. And in fact, this interconnection is from the both direction, we can say. And but this has also some disadvantages, in the sense that these breeds are also particularly sensitive to possible climate change occurring in their own environment if they are marked and quickly, and especially in case of, for example, extreme weather events that could easily cause irreparable damages to specific population living in those regions. So I wouldn't talk too much about this because previous presenters did it very well, but genomics is now recognized as a pivotal tool to face the challenges of the future climate change. And the last decades have seen incredible advances in this field with enhancement and developing new technologies paired with a remarkable decreasing cost of genotyping. And so a very large number of national and international projects that produce huge amount of genomic data about different plant and animal species, including sheep and goat, have been observed. And genomics can be used both for looking for genes that account for the phenomenon of environmental adaptation, as we've seen, but also, for example, to preserve the biodiversity and the genetic variability both within and among different breeds. And this is aimed at the selection, an animal selection that wants to balance productivity and sustainability to produce in a very sustainable way in the future. So today we would like to present you at first a brief summary of a work we developed with the Italian Goat Consortium that we published last year. in which we explore the genetic structure of Italian goat population with respect to present and future climate conditions of their respective breeding range. So the dataset we used came from the Italian Goat Consortium and is, as far as we know, the largest national level genotyping effort about goat biodiversity, including over 1,000 goats of 33 Italian breeds plus a wild species. All the animals were sampled with a medium density SNP chip, and the samples were all geolocalized so that we could obtain environmental data about the different location of the breeds and of the samples. First of all, we analyzed the population structure of all the breeds. Here you can see the multidimensional scaling plot, in which we excluded the Monte Cristo goats because they were quite isolated from all the other breeds, so the visualization was not optimal. But in this case, you can see that the first component clearly highlights a north to south geographic gradient. And this is confirmed by the admixture analysis, in which when we used 3 clusters, we could clearly differentiate breeds from the south, central south, and from the northern Italy. In particular, this reflected also the socio-political situation that preceded the Italian unification. In fact, you can see, for example, that the yellow cluster predominates in the northern breeds that were part of the Sardinian kingdom, whereas the blue cluster that was led by the Girgentana breed, Sicilian breed, you can see it here, characterized the central southern breeds that were part of the kingdom of the Two Sicily. And lastly, the purple cluster was characterized the Maltese population and the derived breeds. These 3 clusters were also reflected in the haplotype sharing. So you can see, for example, that the northern breeds showed a larger haplotype sharing in Lombardy than all the other alpine regions, and that there is very small sharing with other southern breeds. Instead, the central southern breeds show a larger haplotype sharing both within and among different clusters, including, for example, the insular and Maltese populations. The second part of the study regarded the landscape genomics analysis, which were carried out using, as we see before, Sambada and LFMM approaches, and that was done in collaboration with the University of Piacenza. So, passing to the results directly, we could identify about 220 SNPs falling on 62 different genes that were associated to these 4 main, most representative environmental variables. And going to analyze the biological role of these genes, we could classify most of them in one of these 4 groups that are methane growth, circadian rhythms, fertility, and the inflammatory response. In particular, it was very interesting that one of the genes in these last groups can be considered candidate genes for the response to the caprine encephalitis arthritis virus infection, because it is associated also to the infection, to the response to the HIV that is part of the lentivirus family as the virus causing the CAIV. Lastly, the last analysis aimed to predict in a very simple way how the allelic frequency of this gene might change in the future. So at first we used the Beck et al. Data to group breeds according to the Köppen-Geiger climate classification of their breeding range. And for example, we observed that most of breeds at present live in temperate regions, but that about 2/3 of them might change their Köppen classification within 70 years, going to live in more drier and hotter regions and climate. So we created this very simple model, dividing the breeds that at present live in hot or not hot and in dry or not dry regions, and applying a one-way ANOVA to verify if what SNPs had an allelic frequency that differentiated these two groups. And we found that 38 SNPs could do this. 9 of them were fell directly on one annotated gene. So we applied on these 9 a linear regression model that used their mean frequency, allelic frequency, as well as the mean value of the associated environmental parameter. And we found out that this model was significant for 5 of these SNPs. So we could, using the delta of the 2 parameters, we could predict the possible future allelic and genotypic frequency of these genes, and we saw that some of them might go towards stabilization or fixation. So now we come to our latest work, which focuses more on the Italian sheep and goat population and related climatic threats. We consider this topic particularly relevant, as demonstrated by the fact that the FAO includes it among its strategic priorities, because not only have a deep knowledge and accurate knowledge about the real localization of a breed in its total, in its whole, can allow to have accurate geographical information, environmental information, and thus identifying genes that are associated to the adaptation to that environment. But it's also important to monitor the risk status of the different breeds, because as I mentioned before, there are some breeds that live in very small areas, in very little areas, and so some possible catastrophic events could easily wipe out these breeds. Thus, for this work, the National Breeders Association of Sheep and Goat, that is ASSONAPA, provided data about farm size and localization, as well pedigree data about 34 goat and 42 sheep breeds from all over the peninsula. We also gathered publicly available genomic data of 25 goats from the Italian Goat Consortium that I mentioned previously, and from 18 sheep breeds from the BioVita project, all coming from medium density SNP sheep. And then we created some maps using different environmental and climatic parameters. And in particular, we used again the Copenhagen classification from Beck et al. For present and future. And data about the district level risk of landslides and flood by the ISPRA website, and the seismic classification at district level from the Protezione Civile website. And the Protezione Civile is practically the Civil Protection Corps of Italy. So for each breed, we created a distribution map in which data were aggregated by province for visualization province. And so you can see, for example, here the distribution of the Camosciata delle Alpi goat and the Merinizzata Italiana sheep, in which each circle corresponds to a province in which the breed is present, and its radius, as well in this case as its color, depends on the number of subjects present in that province. With regard to the landslides, the flood risk maps, we had a table in which each district was associated to a percentage of area that was at mild, severe, or moderate risk of landslide or flood, obviously. But we simplified it using the percentage of the total area at risk. However, in the future, we would like also to account for this different risk level of each district. And lastly, we had a table with the seismic classification of each district. So the creation of this earthquake risk map was quite straightforward, more than the others. So first of all, we analyzed the breed pedigree and calculated their effective size. Using this parameter, it is in fact possible to predict a population risk of extinction, because when the effective size is below 50, the breed can be considered a short-term risk of extinction, whereas when it is between 50 and 500, the population is considered at long-term risk of extinction. So according to our data, we observed that about 21% of the goat breeds and 14% of the sheep breeds were at short-term risk of extinction. But when we consider the longer period, almost, or better, over 70% of these the small ruminant populations were at risk of extinction. Moreover, we observed that most of the breeds live in a very little breeding range. This was expected since actually most of the breeds take their name from the place they developed. But our data clearly demonstrated it. And in particular, we observed that 1/4 of the goat breeds and 1/3 of the sheep breeds are all at short or long-term risk of extinction live only in 1 or 2 adjoining provinces. In particular, here you can see the maps in which we plotted the breeds by province according to their effective size. And for example, you can see the sheep breeds that are at short-term risk of extinction are all concentrated in the northern part of Italy, whereas when we consider goats, there are breeds both in the northwestern part and in the central south of Italy. However, this information tells us that when a catastrophic event could happen in a particular province, some entire breeds could be severely damaged. Using genomic data, we then calculated the runs of homozygosity based inbreeding coefficient, FROH, based on the Macmillan formula. In particular, we observed that FROH was higher in goats, about 8%, than in sheep, about 6%, but we could not detect any difference among the risk classes in terms of extinction. Even though we have to say that unfortunately we had no genotyped sheep breed that were at short-term risk of extinction. Moreover, we also calculated the FROH based on the ROH length, because it is known that the shorter is the ROH, the older is the inbreeding event. So we were particularly interested in the longest class, which corresponds to over 16 megabases, because it is an index of recent inbreeding. And we observed that about 1/3 of the total inbreeding is due to this class. However, again, no differences among the different risk classes was detected, probably because the variability was quite large. Coming to the environmental risk, I will show you some examples of the breeds that stand out for being particularly critical. And with regard to the predicted climate changes, we observed that about 1/3 of the goat and half of the sheep breeds almost completely live in provinces that are going to change their copper climate classification within the next 70 years. And so they will have to cope with hotter and drier climates. In particular, the Garganica goat is a breed that is reared in Puglia. When we observed that we can expect that the arid steppe climate will change from cold to hot, and the temperate climate that is in green here and then in yellow here will become from not dry to dry. So this is the confirmation of the general observation. And moving toward the north, we can find, for example, the Sopravissana sheep that live in the central part of Italy. And this breed has already experienced some great problems due to the earthquakes that hit those regions in 2016. But looking at these maps, we can expect that unfortunately it will have to face other climate challenges also in the future. As for the Lelange sheep, it is a sheep breed that is bred in the western part of Piedmont. And we can see that the alpine region is particularly critical, as demonstrated by the fact that the increasing in its temperature is double with respect to the global means. And we can also see here that we will lose a great part of the cold and polar area all over the Alpine region, both in Italy but also in the rest of Europe. In this slide, you can see the risk of landslide, as I said before, expressed in terms of the percentage of the district area at risk. And in particular, I would like to show you, for example, the Cilentana Nera goat, that, as well as the other two Cilentana goats, is only bred in the southern part of Campania. And you can see that all this area is completely at risk of landslides. In the northern part, instead, we can find, for example, 3 different breeds, sheep breeds that might be accurately monitored. And in particular, we have the Garfagnina sheep that is bred in the northern part of Tuscany, that is also at high risk for flood, as we will see later. The Rosé sheep that is bred in Val d'Aosta, and the Schwarznasenschaf that is bred only in the province of Bolzano. You can also observe that all these breeds are also at long or even short-term risk of extinction. As for flood, we can see, for example, the Garfagnina goat that, as you can imagine, live in the same area of the Garfagnina sheep in the north of Tuscany, but the region that is at higher risk for flood is the Emilia-Romagna, where, for example, the Cornilio sheep live. The last maps I'd like to show you are about the seismic classification of the Italian districts, where the class 1 correspond to the higher risk and interest almost all the Apenninic, southern and central region. Going to the southern regions, we can find, for example, the Nicastrese goat that lives in Calabria, but in this same area also the other 2 breeds, the Aspromontana and the Rustica di Calabria, are present. So an eventual earthquake in this region could easily cause irreparable damages on at least 3 small ruminant breeds. Instead, if we move to the north, we can find, for example, the Bagnolese sheep that is bred in this region. So in conclusion, from this work, we observed that most of the Italian small ruminant population are bred in limited areas, and that about 1/3 of these breeding ranges are also at risk of being hit by a natural disaster or climate change. This information is obviously to be integrated with, for example, the data about the effective size and the inbreeding of these populations. But even if these are just preliminary results, it emerges that further research about the accurate identification of the distribution of the different livestock local breeds is needed in order to assess what are the breeds that needs to be carefully monitored, and for what breeds we should maybe develop and implement some breeding, some conservation and selection programs. And obviously there is still much work to do, and we hope also to collaborate with some of us if you would like to increase the knowledge about this. But what we plan to do in the very near future is to create a multivariate risk index that includes all the parameters that we have seen before as well as other. For example, we would like to include more environmental threats like wildfire, droughts, or other environmental parameters, such as I'm thinking about the slope or the land coverage of different vegetables, but also including, for example, information about the farm structure, so different husbandry practices, or, for example, the age of the breeders. That can be important because some breeds are bred only by old people that I don't know how long, unfortunately, could be with us. And for example, other agriculture system, agro-territorial services that these farms provide. And for example, also the contact with other species, because these breeds might have problems of health because of the contact or predation or hybridization. And finally, we would like to increase the number of geolocalized genomic data for all the different breeds, so that we could also create maps of the genomic inbreeding, for example, and have all the information needed for further investigation and the analysis, the landscape genomic analysis, obviously. So thank you, and if you have any question, I would be glad to speak.
So another great presentation. Thank you, Arianna. Do we have any question from the audience?
Thank you very much for your presentation. I have just a curiosity. How can the earthquake can affect for example, the biodiversity of goats? I mean, because do you have information from farmers that these goats, after the earthquake, they die, they collapse in a bottleneck, or not? Because for me, it was a kind of surprise that there is a earthquake effect inside climate change. Thank you very much.
Thank you for the question. We had some problems, for example, with two breeds that are the Sopravissano sheep, that I mentioned before, and the Terramana goat, because of the earthquake that hit the central part of Italy in 2016, because a lot of farms were destructed practically, and also people think that have some problems. For example, they have to move or something like this. So there are examples of a reduction in the population, not only from Italy, but also, for example, I think from the Kenya earthquakes that happened some years ago. And you can really see a completed fall in the population. So it depends on the region that it eats. But if it's a province that is the only province in which a breed is reared, it might be possible that a great part of the farms may be destructed or have severe consequences, also from a structural point of view, maybe.
Thank you. Are there other questions from the audience?
Maybe, so I saw the estimates of effective population size. So this could be a problem for inbreeding. Then there are some papers coming out in these recent years, and they say that you can have a low effective population size and still be okay, because it depends on the amount of deleterious mutation and mutation load. They say if you have a low effective population size for a long time, then you have purged your deleterious mutation, and the problem is when you have a declining populations. And so I don't know if in these samples you could estimate this mutation load and see if there are some varieties that have a higher mutation load or not, and or also that problem of the earthquakes. When you have really a bottleneck, that could be really a problem in that.
Yes, sure.
So it was just a reflection.
Yes, sir. I agree. And at the moment, these are preliminary results, and we use just the traditional formula based on pedigrees. So the ratio between males and females, and with the appropriate correction. But taking new data, and we intend to do it in a national project which aims to samples almost all these breeds in different locations, and maybe we would apply the target sampling, going to samples from also different environments and different towns and provinces. We might also include all these problems and this data in future risk assessment, for sure, from a genetic point of view.
Okay, thank you. And the other question is just on census size. So when there is a herd that is maybe 50 individuals, so it's very small, would it be possible to increase the number by?
This is, I think, a very difficult, a very discussed problem. For sure, what we can do is at least conserve this small, yes, this size. For example, with ex situ and in situ conservation programs, some breeds maybe will have to have some crosses between different breeds, even if obviously some biodiversity would be lost, maybe. Some other breeds might be instead used in environments that are expected to change and become similar to those in which these breeds are developed. For example, I'm thinking about some Sicilian goats that maybe are adapted to a more arid and hot climate than others. And if we see that another place in Italy, or even not in Italy, is becoming similar to this environment, in its environment to Sicily, maybe we can propose to expand the breeding range of these breeds, because maybe they might produce more in the future then, I think.
Okay, so if there are no further questions from the audience, thank you again.
Thank you very much.
And I think Licia has some concluding message for the day.
Okay, so as you may guess, the first day of the workshop is over. And now, on behalf of the organizing committee, let me thank again all the speakers that today really brought excellent contribution to our general discussion, our main lecturers who provided a very nice and comprehensive framework of the topic and of the applications with by revising a number of methods that I think most of you will find very useful for future application in your own fields, and also our chairman, Marco Andrello and Oliver Silmoni. Thanks again, and see you tomorrow morning for the second day of the workshop. Thank you for attending, and also thanks to who attended online.